In the endoplasmic reticulum, the MHC class I molecule consists of a heavy chain and a light chain, which will be added later on. In order to prevent peptides from binding to the MHC molecule, first connexin is bound to the molecule. Secondly, calreticulin will bind, instead of connexin, to the MHC. And this causes the association of the beta-2 domain and other proteins such as ERB57 and tepsin. The MHC complex consequently binds to the TAP transporter via tepsin. When a pathogen enters the cell, it will start making its own BAP proteins. Consequently, these proteins are degraded into protein fragments by the proteasome, which happens in the cytosol. Via the TAP transporter, the protein fragments are able to enter the ER. There, the ERA protein will bind to the fragments, which consequently enables the fragments to bind to the MHC molecule. These peptide MHC class I complexes leave the ER for presentation at the cell surface. The affected cell will present the peptide to CD8 T cells. First, the CD8 receptor binds to the MHC molecule to check whether it is really an MHC class I molecule. If this is correct, the T cell receptor binds to the MHC molecule, and in the end, the CD8 T cell will release chemical factors in order to kill the infected cell. Via phagocytosis, a pathogen can enter the antigen-presenting cell. Lysosomes having an acidic environment will fuse with the pathogen and a phagolysosome will be formed. Due to the acidic environment, the pathogen is broken down into peptides. Within the ER, the MHC class II molecules are synthesized. The invariant chain will bind to the molecule to prevent proteins in the ER from binding to it. The invariant chain also acts as a chaperone, bringing the MHC molecule out from the ER by endosome formation. This endosome is consequently transported through the Golgi apparatus in a new endosome. This new endosome is slightly acidic, which results in degradation of the invariant chain, leaving a fragment called clip. Another protein called HLA-DM will interact with the MHC molecule, releasing the clip. The phagolysosome will then fuse with MHC class II endosome, and this will allow the peptides to bind to the MHC class II. Consequently, the APC will express the MHC class II complex on its surface. The MHC class II is expressed and will wait for interaction with an naive CD4 T cell. When there is a CD4 T cell present, the CD4 receptor will check the MHC molecule for its class. If that's correct, the TCR will bind to the MHC molecule to check the antigen. This will lead to an activated CD4 T cell.